Now we will look at imperfect fungi and lichens. Deuteromycotina is an important division of fungi with around 17,000 species. However, it is a dustbin class. Into it are thrown species that have no known normal sexual stage, so that they cannot be placed with confidence in other classes. Most reproduce by conidia, although a few are purely mycelial, developing no spores. Their mycelium is state-based and profusely branched state-based. The majority of the fungi in this class are likely to be ascomycetes, in which the ascus stage has not yet been discovered, or in which it has been lost in the course of evolution. A few may be conidial stages of basidiomycetes. Theoretically, when, as frequently happens, the ascus stage of a conidial fungus is discovered, it should be transferred to Ascomycotina. However, it is usually convenient to retain such a fungus in Deuteromycotina if the asexual stage is the one usually to be found in nature. Parasitic deuteromycetes are the causative agents of disease of plants and animals, including humans, and some cause spoilage of stored products. Deuteromycotina, a representative genus of Deuteromycetes, is Fusarium. Fusarium species are cosmopolitan in soils, from the permafrost of the Arctic to sand in the Sahara Desert. They are particularly common in cultivated soils, are associated with a wide range of plant diseases, and are important mycotoxin producers. Some species are also known to cause disease in humans, such as nail infection, keratomycosis of the cornea, ulcers, necrosis, skin infections, and fatal infection of internal organs, especially in immunocompromised patients. Species of Fusarium typically produce both micro and macroconidia from slender phyalides. There are structures like a branching conidiophore. Microconidia are two or several cells sickle-shaped, and macroconidia are one or two cells smaller than the macroconidia. A representative Another group of anamorphic fungi are known to be nematophagous. Fungi and nematodes being both highly present in soils, some nematodes prey on fungi, but it can be the way around. Fungi have evolved a range of mechanisms to attack nematodes, which can be grouped into three broad categories: predatory, endoparasitic, as well as egg and cyst parasites. Species like Arthrobotrys conoides are predatory and produce a sizable mycelium in the soil, with trapping devices formed at intervals along the length of the hyphae. The most common traps are sticky knobs, adhesive networks, non-constricting rings, or constricting rings. Penetration of the captured nematodes is followed by the growth of the trophic hyphae throughout the nematode's body and digestion of its contents. This fungus reproduces by means of two-celled pear-shaped conidia, in which the cells are of an equal size, with the smaller cells nearer to the attachment points of the conidiophore. We will now look at lichens. A lichen is now defined as a self-supporting association of a fungus, also named Mycobiont, and a green alga or cyanobacterium, identified as Photobiont, resulting in a stable thallus of specific structures. The fungal partner usually contributes most of the biomass to this symbiosis, including the external surface. It is thus termed as the exhabitant, whereas the unicellular or filamentous photobiont cells are collectively called the inhabitants because they are located inside the lichen thallus. Most lichens have a characteristic appearance, which permits their identification. About 85% of lichenized fungi have a green algal photobiont, and 10% are associated with the cyanobacterium. The remaining lichens contain both a green alga and a cyanobacterium. Most lichenized fungi, more than 98%, belong to the Oascomycetes, with only a few imperfect fungi and some 20 species of Basidiomycota also entering this type of symbiosis. We will now look at lichens. Since the structure of lichens is almost entirely due to the fungal partner, lichen taxonomy is synonymous with the taxonomy of the Mycobiont. Some 13,500 species of the lichenized fungi have been described to date. Since lichens are often conspicuous and have been relatively well researched over the past 200 years, this number is not far below the estimated worldwide total of some 18,000 species. Some lichen thalli live for over 1,000 years and can be used for determining the age of rock surfaces because of their slow growth rate. This discipline is known as lichenometry. It has been applied, for example, to date the standing stones on Easter Island or the time point of exposure of rock surfaces caused by avalanches and earthquakes. Because of their ability to tolerate repeated cycles of drying and dehydration, and to survive extreme temperatures, high solar irradiation, and other adverse conditions, lichens can colonize a range of terrestrial habitats compared to flowering plants. Lichens occur in all climatic zones, from the Arctic and Antarctica, where they provide the dominant vegetation, to the tropics. Lichens are classical pioneer organisms and can be found on bare rocks or in fertile soils. They can also cause the weathering of rocks by secreting oxalic acid, which reacts chemically with the rock surface. Extensive lichen communities also exist on the bark and foliage of trees, known as corticolous lichens, and can also be found on animals, like here on the shell of a turtle. Additionally, freshwater and marine species have been described. They are rare in polluted areas, being intolerant of industrial pollutants, especially sulfur dioxide. In this relationship, the fungus gets organic nutrients from the photosynthetic activity of the algal symbiont. It is possible to grow the algal and fungal partners of many lichens separately in pure culture. Whereas most photobionts multiply readily in pure culture, the fungal partner, if it grows at all, typically shows slow growth with a sterile leathery mycelium, but does not produce the characteristic lichen thallus. That means that with their photosynthetic partner, lichenous fungi can live longer and fulfill their sexual and asexual cycle. In this relationship, the fungus gets organic nutrients from the. On the other hand, The photosymbiont receives mechanical protection by the fungus from adverse environmental conditions, such as high solar irradiation, by being tiny enveloped by the hyphae. This translates by the bright colors of the lichens due to the presence of cortical pigments which screen out UV light. The fungus allows better retention of water and thus improves the resistance to desiccation for the photobiont. All those conditions provide a great environment which permits the growth of the photobionts in situations which might be too hostile for free living forms. A lichen consists of a simple photosynthesizing organism surrounded by filaments of a fungus. Below the cortex layer is a layer called the photobiontic or symbiont layer. This symbiont layer has less densely packed fungal filaments with the photosynthetic partner embedded in them. The less dense packing allows air circulation during photosynthesis, similar to the anatomy of a leaf. Each cell or group of cells of the photobiont is usually individually wrapped by the hyphae and in some cases penetrated by a hostorium. Exchange of nutrients is then made through wall-to-wall contact between the two organisms. Lichen thalli come in three basic shapes: crustose, which is crust-like, fruticose, that are shaped like a miniature shrub, or foliose, more leaf-like. It should be noted that these are purely descriptive terms, which have no taxonomic meaning because intermediate forms also exist. Lichen thalli. By far the most common type is the crustose thallus, which forms a thin, spreading crust firmly attached to the substratum by its entire lower surface. In the morphologically simplest crustose lichens, fungal hyphae are loosely associated with photobiont cells, but do not form a protective upper surface. Such lichen thalli appear powdery and are referred to as leprous. They often have highly hydrophobic surface. Other crustose lichens produce a thicker thallus, often held together by mucilage, as in the gelatinous lichen. In more highly differentiated crustose thalli, the photobiont cells are positioned in a defined layer located underneath an upper cortex formed exclusively by the mycobiont. The photobiont cells are thus protected from adverse environmental factors. Air spaces in the photobiont layer and the medulla underneath permit gas exchange. The differentiation of the lichen thallus into horizontal layers is called stratification. In squamulose lichens, the crustose thallus forms small scales which become partially raised from the substratum, giving the surface a scruffy appearance. The stratification is developed further in the second thallus type, the foliose lichens, by the development of a lower cortex. Attachment to the substratum is often by bundles of hyphae termed rhizinae or rhizines. As a result, the thallus appears leaf-like or lobed and can be detached from the substratum without being damaged. The third thallus type is called Fruticose. Here the thallus has a shrub-like or branched appearance and is raised from the substratum or hangs down from it. In some cases, a fruticose thallus may develop from a basal crustose or foliose thallus. Stratification in Fruticose thalli is often tubular rather than horizontal, and it resembles the more complex Crustose types in possessing an outer cortex overlying a Photobiont layer and a nucleus, but lacking an inner cortex. Sexual reproductive features of lichenized ascomycetes are largely equivalent to those found in non-lichenized fish. Both homothallic and heterothallic mating systems are known. Apothecia with inoperculate asci are especially common, but perithecia and pseudothecia also occur. In contrast, no Pleistoctesia or apothecia with operculate asci are known among lichenized fungi. In most cases, the photobiont is excluded from the fertile regions of the mycobiont so that the ascospores or conidia are released without photobiont cells. A new lichen thallus can be established from fungal spores only by re-lichenization with suitable photobiont cells. Most lichen thalli also produce vegetative propagules containing both symbionts. They can be very variable, and an extensive terminology has evolved to describe them. The most common vegetative propagules are zooidea, which are small clumps of hyphae enclosing a few algal cells. They are produced over the entire surface of the thallus or in differentiated structures called soralia. For this lab, you will have to look at a descriptive table of two cultured specimens, Fusarium and Arthrobotrys. You will also have to look at the material available to you and make the 7 following drawings of Fusarium, Arthrobotrys, 2 foliose lichens, 1 Fruticose lichen, as well as 1 sketch of a Crustose lichen specimen and 1 of the foliose specimen Physica.
Okay, so just one quick note about the pre-lab. When you'll notice that there's an extra section for your table, the conidiophore branching, we just are looking for if it's branched or not. And then, so you'll have your lab 9 pre-lab is on D2L, and that's going to be do the same, it'll be like your lab assignment. So it'll be due the day before, the day after next week when we have our lab session. We'll still have a mandatory lab session. And yeah, so that's available to you now. So if you have any questions about that, feel free to ask.